So, hi everybody. My name is Mark Sendak, and as was just said, I'm here representing the Duke Institute for Health Innovation. So, I'm going to be speaking a little bit of background about our team, the way that we're structured, and then kind of walk through how we have iterated our process and practices over the last 8 years, and then talk about how we look for external partners in the work that we're doing. So to kick things off, our mission is to catalyze innovations at Duke. Every project that we do typically tries to address a clinical or operational problem within the care delivery setting. And our approach emphasizes design. So we work with end users on projects. Every project has frontline workers directly involved, and we work with them to design and develop solutions to problems that they face every day. We have two different ways that we identify problems. On the left-hand side, I'm going to talk a little bit more about this later in the presentation. It's our request for applications. And then on the right-hand side, this is kind of an innovation competition whereby we work with faculty who have built something in a laboratory environment, and they're looking to commercialize the technology and take it to market. We try to push the boundaries of what is possible rather than thinking about moonshots, which are very high cost investments. We think about roof shots or slingshots. These are lower cost investments, but can have significant return within the health system. We also try to build products and services that allow more folks across the organization to do innovation within their own parts of the organization. And lastly, we are really all generalists at our team, DHI. We work across the care continuum, across conditions, chronic disease, acute illness. And I'll talk more about different aspects of this, but we've really built out a whole platform of education programs and technology infrastructure to enable our projects. So next, I'm going to kind of talk through the different principles and practices. So we break it down into 4 principles: build to show value, build to integrate, build to scale, and build responsibly. And I'm going to go through each one of these bullets and give concrete examples and how they relate to our projects. So probably most importantly, when you're building new products or services, is to make sure that you're working on the right problem. So one analogy that I often use when talking about doing innovation in healthcare is that we're laying the train tracks for the train. So a health system is a very large organization. Duke has about 30,000 employees, and we move slowly. And it's our team's job to be looking ahead of the organization. Where do we need to be going? What does the landscape look like? Where are there opportunities? And we lay the tracks, we flatten the ground, we help the train go on the tracks, but then we need to get off the train and continue kind of looking ahead at what's next. Another way to think about the type of opportunities we go after. So this is called the innovation value chain. Starting from the left-hand side is generation, and then conversion and diffusion of innovations. It is not our job to come up with the ideas. The ideas are really being generated on the front lines of the organization. So these are clinicians, pharmacists, administrators, people who are managing the delivery system. They have ideas about how to improve their work. We get involved to do the conversion. We select projects, and then we develop the first version of a solution. And I would say that we often are asked to participate in diffusion. Sometimes we support scaling a project, but that's not an area that we focus. And if China is anything like the US, health systems are very diffuse. And if something is successful in one setting, it doesn't mean that it automatically gets diffused across the system. So that requires kind of a different set of relationships and resources. And lastly, we focus on transformational and adjacent opportunities. So in our own system, we have entire kind of teams and capabilities around performance improvement, and we view those as core innovation. So you're optimizing existing services for existing patient populations, whereas when you're trying to reach new populations or creating new markets, those are adjacent or transformational opportunities. Next is something that we spend a lot of time thinking about, which is how to align the frontline staff to the senior leadership priorities for the health system. So this is what our request for applications looks like. So starting at the left-hand side, the C-suite, so the chief executive officer, chief medical officer, chief financial officer, a small team of executives, they identify 4 to 5 strategic priorities for the entire system every year. And we work with those leaders, as well as all of the leaders of different business units, to publicize this proposal opportunity. Frontline workers then submit proposals that are reviewed by the business unit leaders and the executives. Once projects are selected, we work with business unit leaders and the frontline workers throughout a 12-month period to design, develop, and implement solutions. And then we have regular touch points with the senior executives to provide updates on the project. So we really handle a lot of the communication across different levels of the organization, and then we rapidly iterate on the product and service development. Next is the idea that we have to really create synergies between the management and leaders. And these are often different skill sets and maybe different individuals. When you're trying to do something new and really create change within the organization, identifying who can motivate people, who can align people, establish a direction for people to go forward on. So a lot of our time is spent just doing stakeholder engagement. The last concept and practice within Build to Show Value is starting with minimal viable solutions. So this comes from the lean startup methodology. Don't let the perfect be the enemy of the good. This is often a struggle in an academic setting like Duke, where we have a lot of faculty and experts who want to understand the best way to do something before they feel comfortable actually using it. Whereas we are an innovation team and we try to solve core problems and then add complexity over time after we start solving the problem. So here's an example of the first version of the prototype for our sepsis watch system. It was initially built in PowerPoint. This was a slide that we put together with nurse leaders and we iterated on with them to help map out the workflow. And over time, that was built into a custom web application that is now used 3.5 years later, primarily on iPads. The next principle is building to integrate. So as I mentioned at the beginning of the talk, all of our projects try to solve clinical or operational problems within the organization. These are things that are meant to be translated into practice. And we're not a research team that's just trying to publish papers. It has to be integrated into the system. So it's commonly known that a lot of these technologies are built without strong engagement of the frontline clinicians, and they're not really centered around the development and not meaningfully trying to figure out, well, how would this be used in practice? And that's the antithesis of how we operate. We bring together these different sets of expertise. We have weekly meetings, and we're collaborators in both the invention process and the academic dissemination process. One of the challenges we run into, though, is that these different domains of expertise are often siloed within our organization. We have the clinicians who are part of the health system, we have faculty in the departments on campus, and then we have our IT organization that manages the electronic health record. So what we've had to do is create the interfaces that allow people across these domains to collaborate and work together collaboratively. The next piece for building to integrate is that you have to staff projects and you have to provide that project management support to allow the project to move forward successfully. There has to be dedicated capacity Frontline clinicians are busy. They already are overloaded and overworked. And at least in the US, especially during COVID, there's been much more significant burnout. So we have project managers, we have full-time staff who help with the technology development, the implementation, the evaluation, and we augment the capabilities that already exist within the system. And lastly, when you're talking about build to integrate, and you're developing a new type of technology that's never been used before within the system, the organization has to be willing to change. So I'm going to give a few examples of cases where we had to create new workflows and new structures to solve the problems. So this is an example of chronic kidney disease progression. We worked with an accountable care organization, part of the health system that manages care for 50,000 older adults. Within these 50,000 older adults, we wanted to identify who is at greatest risk of having end-stage renal disease, so needing dialysis or needing a kidney transplant. So we used a published algorithm that we ran on data for all of these individuals, and we identified about 2,000 people who were high risk. Then we went through and we eliminated people who were already seeing a specialist, already had the outcome that we were trying to avoid, and we found about 400 people in the community who had progressing disease. And we needed to develop a workflow where we were going to connect those people to care, because we ran these algorithms and we identified these people, but they were out in the community and it's not like we were regularly seeing many of them. So what we did is we created a new workflow called population rounding, where we aggregated and curated all of the data. We ran it through our computation algorithms, the normalizing, harmonizing all of the data. And then we brought together an interdisciplinary team to review these high-risk patients and decide on a best care path for these patients. And then there would be nurses making phone calls to the patients, scheduling them with their primary care physicians to carry out some of these interventions. And this was entirely new for the organization. A similar example is how we use our sepsis algorithm, where instead of sending alerts to clinicians in the emergency department, we had done this previously where there was a pop-up that showed up on the screen and 86% of the alerts were canceled and there was no clinical impact. So the emergency department, they asked for help triaging and placing all of the orders to treat sepsis. So we used what we call a patient response program, rapid response nurses, who now provide dedicated support to the emergency department staff. And it's really changed the way that these nurses work. And this is a quote kind of describing how they have a new way of doing their work now in the hospital. Another idea is that you actually need to move pieces of the organization. So this I'm saying kind of as a joke, but in the United States, when you come into the Duke Hospital, you see these plaques. This is the organization chart from Duke when it was established 100 years ago, and it's set in stone and it hasn't changed in 100 years. Whereas when you need to respond to new technologies or new opportunities, you actually need to change the structure of the organization. One example of that is how we responded to COVID-19. So we are part of a big university campus. We had to build a lot of technology very quickly to help with remote monitoring and symptom monitoring for all of our students and staff. But we needed to get those support services out into the community. When we went out and started talking to people in the community, there was a lot of lack of trust of Duke Health. These communities felt ignored. They saw resources going to student programs and not to the community themselves. Really being able to care for yourself during COVID was a burden for people because they had to still go to work. They didn't have the childcare help that they needed. And so when we were thinking about, well, how do we support the community? It really took a very different type of approach. So we created a new entity called Greenlight. And through Greenlight, we were able to support the community and help people identify where they could get help with food, medications, paying for their bills, paying for rent. And this was a tool that ended up being used by businesses like restaurants, cafes. It was used in schools to help schools stay open. And this is another quote from somebody who owns a bar, a restaurant in Durham, about how these tools that we built originally to support the university, we had to take it out of the university and now support the local businesses with restaurants and food services. The next principle is building to scale. So once you've had success in one part of the organization, how do you continue to do these projects and scale the impact? So one idea is this comes from a book called Crossing the Chasm. And it's the idea that when you're doing something new, not everyone is going to want to work with you. And a lot of people are not going to feel comfortable using a new product or service. But you don't need to target everybody. You only need to target the early adopters or the innovators who are interested and already want to work with you. So here's an example where for one project, we engaged all of these different stakeholders and different experts across the system. And this was a project that now has been running for 3.5 years. And almost every single person who was involved in this project has now been involved in other things that we've done. And they've brought other ideas to us. Because as people start to see success, it starts to get them thinking about, okay, if AI or machine learning works for sepsis, maybe it'll work for cardiogenic shock. Maybe it'll work for mortality. And these are just 3 different examples of people who have become evangelists and come back to us every year or every few years with another set of ideas to work on within the organization. And then part of scaling is also making sure that you have the infrastructure to support a growing number of projects. And the infrastructure has to be modular. So this is just a schematic describing our data pipeline. It uses a lot of open source tools, Airflow, Docker, Kubernetes, and it's an entirely automated pipeline that extracts data from our electronic health record, which is Epic, normalizes the data, and it allows us to run many different algorithms, populate registries, present data and model output on dashboards to users, and driving workflows within the health system and to do quality reporting and monitoring. So I'm just going to show you a few examples of what the dashboards look like. These are mostly dashboards that we've built in Tableau. So this is one dashboard that's used to look at 30-day mortality in one of the community hospitals. This is a dashboard that's used in the outpatient setting in clinics to identify patients who are likely to have non-alcoholic fatty liver disease. This is a dashboard showing every bed in the emergency department and overall how many patients in the emergency department at any given time are high risk for needing an ICU bed, like critical care, or an intermediate bed. So this helps with capacity planning when you're trying to figure out how many beds you need to have available for people. And when COVID hit, we were very quickly able to use a lot of these tools to do COVID monitoring and monitor ECMO, monitor ventilators, how many patients had COVID on every one of the wards. So this shows every bed in the hospital. And when you're running algorithms, you also have to monitor the data very closely. So we do data monitoring for everything that goes into a model. And the last piece of scale is the human capital. We are an organization, as I mentioned, with 30,000 employees. There's many, many experts, many, many frontline workers, and we want to build those capabilities. One thing to be aware of when you're building AI or machine learning is you're integrating these technologies into complex organizations. So we often hear people use the word deploy, but we are very thoughtful of saying, well, we integrate these tools into workflows, into professional relationships, into existing norms within the organization that we have to understand. Another piece of this is that this is similar to crossing the chasm in how you identify evangelists. As you start to have success in the organization, it plants a seed. Other people see what's possible, and other people start using the tools and using the approaches to do work in their part of the organization. And lastly, we are a big university, so we run a lot of training programs as well. So in any given year, we probably work with 100 trainees at least. The last few things I'm going to touch on are best practices for building responsibly. For every model that we build, we create a model label. So these, we've called them model facts labels. There's also model cards, there's different examples of this. We do a lot of data quality assessment, and this isn't something that you just do once at the beginning of a project. It's something that needs to be continually monitored after a model is being used in practice. This is a big monitor we have about 60 inches in our office, where it shows the sepsis dashboard and it shows the monitoring for all of the input data elements. And I already showed this example of we monitor all the data that goes in. So just to kind of quickly recap, build to show value, build to integrate, build to scale, and build responsibly. The last thing I'm going to touch on briefly is how we look for external partners to help scale impact of the work that we're doing. So this is a literature review that we published a few years ago about how these new machine learning technologies are translated into clinical practice. And being one organization, we're able to help integrate these systems internally. But the reality is, if you see something that works for a condition in one setting, the next step is to externally validate it. See how well it performs in different types of patient populations, and then to diffuse it into other environments to help drive adoption, to do the continuing monitoring and maintenance. We're doing our first model update this year for a sepsis project, but this is an area where we are seeking external partners to help understand how to do this work better. The other is that we often work with social scientists who do field work at our site to help surface the best practices. How do humans use these technologies? How does it change the relationships that they have with each other? How does it change the structure of the organization? That way, this helps us better understand why things work, why they don't work, and how we need to keep improving the organization. So thank you for inviting me. These are the pictures of all of my colleagues. It's a pleasure to be able to share our experience, and I look forward to answering any questions people have.
Hello, it's my great pleasure to be here with you today. And I'd like to thank to the organizers for organizing this wonderful meeting. So today I will talk about infusing structure and knowledge into biomedical AI and what makes this so critical to advance scientific discovery and applications of AI in medicine. The code and databases, data resources, and papers that I will highlight throughout the talk are all linked from our lab website. So if you go to zitniklab.hms.harvard.edu, you will find many pointers to the resources mentioned throughout the talk. Biology and medicine crucially depend on existence of various instruments. And while in the past, those instruments were primarily physical devices, such as microscopes that have facilitated the discoveries, we now need instruments for modern data-intensive sciences. And I envision that those instruments will in a vital way depend on knowledge discovery from data. They will not be physical devices anymore, but in many ways we are seeing how virtual devices or algorithms and AI-driven tools can help us guide scientific discovery and address various problems in medicine. But what makes applications of AI in biology and medicine so unique and so challenging and distinct from the other uses of AI? So I would argue that there are a variety of reasons that many other speakers have already addressed. Those that are of particular interest and relevance to this talk are the following. For many problems in biology and medicine, we need to integrate very heterogeneous confounded data that span from molecules all the level to the society. That kind of data often does not fit into a one large flat matrix-based data set, which is a predominant input from many of the machine learning and AI applications algorithms nowadays. Instead, what we need to do is to be able to bring together the data that spans multiple scales, ranges from molecules to the level of individuals and to the level of entire populations that might be heterogeneous and encompasses experimental readouts, curated annotations, or self-reported measurements, and also might be confounded and biased in various ways, as it can be generated by different technologies, measured platforms, or be collected in a variety of different ways. So we need techniques and methods that are able to operate on that kind of multi-model, multi-scale, multi-level data representations. Second important reason, second important property of many biomedical problems is that they really require capability to produce predictions that are not only point-wise estimates of a certain label, but that those predictions need to be actionable hypotheses. They need to translate directly to hypotheses that can be tested in downstream experiments in the wet lab or that can guide decision-making in clinical settings. So as a consequence, this means that optimizing models for accuracy, which is what most today's techniques do, is no longer enough. Instead, we need to move beyond measuring accuracy alone and consider other aspects of deciding when the algorithms perform well and what kind of characteristics they need to have, as they need to be able to sometimes justify their decision-making, produce predictions that are fair or non-discriminatory, and help guide downstream experimentation. So really the question then becomes how we can move away from that kind of black box representation into more white box representation. So to tackle these challenges, I will postulate that a very flexible, unifying way to represent heterogeneous biomedical data is to represent it in the form of interconnected systems or networks or graphs, as we would refer to them as machine learning. Networks and graphs are a unifying mathematical language that allow us to model objects at various different levels, from the level of the genome and genomic regions and how they interact and codepend on each other, to the level of molecules, how they interact with each other, how they are targeted by chemicals or various kinds of therapeutics, how all those systems coexist within an individual with possibly multiple diseases and phenotypes at the same time, and that individual is embedded in a larger local and finally global environment. So networks and graphs allow us to bring that kind of data together and constitute a unified data representation. So they can address this challenge of modeling heterogeneous, possibly confounded, multi-level and multimodal data sets. Once we have the data representation, the question then becomes how we can learn and reason over it so that we can derive predictions that are not only accurate, but also trustworthy and interpretable. And so that is where the notion of infusing structure and knowledge about these interconnected graph structures into biomedical AI algorithms becomes really important. So the algorithmic approach and paradigm that has emerged over the recent few years that has really enabled that kind of reasoning with interconnected biomedical data is known as graph representation learning, or very commonly graph neural networks that specify highly nonlinear and deep transformations on these graph structures in order to produce accurate, interpretable, and actionable hypotheses and predictions. And I will mention a couple of highlights today. That kind of work has enabled applications of AI in a variety of areas, including disease biology, healthcare, and more recently, therapeutics discovery and therapeutic innovation. Some of these applications will be highlighted throughout the talk. Really, the core underlying mathematical underpinning of graph representation learning is the idea of graph neural networks. So in a single slide, this is how graph neural networks would look like. So imagine as our input, we have a graph, which is given to us by a set of nodes and set of edges, indicating relationships, indications between those nodes. Nodes might be a variety of different types. For example, nodes might represent human proteins, might represent also drugs or individuals that interact with each other through familial or friendship or genetic relationships, and might have various metadata and attributes associated with them. But that is our input data. So graph neural networks takes that input network and maps each and every node on the input level into some low dimensional, compact, dense vector description that is often referred to as an embedding. So graph neural network then, here denoted by F, is a nonlinear deep transformation function that will map input structures in the graph, nodes, edges, subgraphs, or entire graphs and represent them as points in some low-dimensional latent embedding space. Crucially, the way that will be done by a nonlinear mapping function F, which is a graph neural network or GNN, is such that latent the embedding space will be optimized such that performing linear algebra operations and that on condensed data summaries or vectors will correspond to capturing the structure and topology of the input graph. And so that is incredibly powerful because it allows us then to go from very diverse, heterogeneous input representations to manageable, low-dimensional, powerful condensed summaries, embeddings that can then feed into a variety of downstream classifiers. So broader implications of that innovation in machine learning in recent years is that with graph neural networks, we can now apply deep learning much more broadly, not only to medical images or biological sequences where we have already seen that deep learning can create tremendous opportunities and advanced science, but we can now apply deep learning to any interconnected complex biological data set. And so this creates some fundamentally new opportunities in biology and medicine, and I would argue will really be crucial for the future of medicine. So let me now walk you through a couple of vignettes that show how we can put graph neural networks into use in order to advance scientific questions. So the first vignette that I would like to focus on is about modeling and understanding therapeutic actions and how the discovery of safe and effective therapeutics might reverse some of disease-associated effects. So it is widely known that many diseases disrupt normal behavior of genes, and so then chemical or more recently genetic perturbations can intervene against those diseases by restoring the function of disrupted genes. However, how to actually restore the behavior of those disrupted genes and how to produce a desirable response in humans that will be safe for them remains mystery for many diseases. So graph neural networks can help in our understanding of how molecular networks that get perturbed in disease, how those perturbations can be reversed by therapeutics. So what I'm showing here in this slide is an example of one type of molecular interaction network, physical protein-protein interaction network, where nodes indicate proteins and edges between them indicate contacts between those proteins in a human cell. So once we have this kind of reference map of the human proteome, we can start asking questions about what kind of interventions and perturbations one would need to design and what proteins in that proteome one would need to target to elicit specific response in patients. And that would correspond to start asking questions about what therapeutic action a novel drug in development or therapeutic would need to have. And we can do that in a cell type specific manner or in a cellular context manner. That allows us to develop AI-based predictors of therapeutic action and patient response. The very exciting part of that work, I believe, is the ability of modeling therapeutic action at the single cell level, where we can start asking questions about how does the action of a particular therapeutic vary depending on the cellular context to which it would be delivered. And those questions can be investigated by constructing a variety of cell type specific protein-protein interaction network, and then in each of those networks prioritizing the list of proteins that are active in that network based on how likely they are to represent therapeutic targets. Similarly, that kind of cell type specific networks can be leveraged to develop predictors of patient response to emerging and incredibly important therapies such as immunotherapies that are characterized by really small sample sizes of today's studies. Yet it's incredibly important to understand why these kind of therapies succeed very well for some subset of patients, but fail miserably for some other subset of patients. So GNNs can help us here construct patient-level networks and then use those networks to identify what are mechanisms of patient response and how likely a particular patient would respond to a therapy. And we can do that even though a very small number of patients might be available to train those models. In addition to working with molecular interaction networks, it is important to be able to ingest and find ways to infuse knowledge about prior biological discoveries and medical knowledge into the reasoning of those algorithms. So to this end, our group has been building a variety of knowledge graphs, including our precision therapeutics knowledge graph, which is a very large heterogeneous graph that brings together information across the entire range of approved drugs and human diseases, as well as information about disease associated and disease perturbed genes, variants, and pathways, as well as therapeutic action of drugs that were approved. In doing so, what we can do is to take precision therapeutics knowledge graph as input and then start asking questions about what is a signature of a particular disease and how that signature, in the sense of subgraphs of genes perturbed in the disease, can be mitigated and targeted by a particular drug. That formulation allows us to nominate candidate treatments for individual diseases. And we are particularly interested in identifying candidate treatments for diseases that have otherwise very few treatment options, such as rare diseases. So here on the right, I'm showing an example of our recent work where we have developed a graph neural network that can be trained on a therapeutics precision knowledge graph, and it can then prioritize drugs based on how likely they are to be effective for a particular disease that has very few treatment options. And what we can see in the second and third group of this plot, we can see that the model yields up to 49% improvement over prior work in identifying treatments for diseases with very few treatment options that currently exist for them and very limited molecular understanding. And the reason why the model RxGNN can do that is because it can really operate on heterogeneous knowledge graph and therefore inject which includes information about prior biomedical knowledge that the model can leverage in a unique way. The use of knowledge graphs and their utility for variety of drug discovery applications and in identifying safe and effective treatments very rapidly is incredibly important in certain situations, such as in situations of public healthcare emergencies that demand safe and effective therapies developed at unprecedented speed. In those cases, traditional iterative development and experimental clinical testing of drugs and approval of new drugs is not feasible. And so the challenge then becomes, well, how can we compress years of work that would take with existing or screening and development strategies into months or even weeks through the use of AI, automation, and new data resources, and very rapidly identify what are promising therapeutics, what are promising chemicals that might be safe and effective for patients. I will highlight that through one particular study that we did recently with our collaborators in Boston, where we were able, during the span of just 3 months, put our AI algorithms to use and train them on a human reference in protein interactome, and then use that graph neural network models to identify what are most promising drugs that could be repurposed for an emerging pathogen. And those AI-based predictions produced by the model were then validated experimentally by our collaborators in human cells. And all that work was done in near 3 months, indicating very clearly how, with the use of AI, new algorithms and data resources, it is really possible to accelerate the process of drug discovery, make it substantially more efficient. So in this particular study, what we were interested in doing is really finding new tricks for existing old drugs. Since the development of new drug from scratch takes on average between 12 and 16 years and can cost between $1 to $2 billion, it's really not feasible in settings where a rapid therapeutic innovation is needed. In those settings, an enticing alternative is drug repositioning or drug repurposing, which is concerned with the question of how we can take existing drugs, old drugs that were previously developed, and whether we can very quickly search through those drugs and identify whether any of those drugs might exert positive therapeutic effect for this new disease that we are interested in. So that is particularly exciting because it can substantially shorten the process of this therapeutic innovation. So with our approaches that we developed that are graph-based few-shot learners, we were able to do that for diseases that otherwise had no successful treatment. So what I will highlight here is just very briefly what few-shot learning is, because it's so crucial for this concept and for this problem. So what few-shot learning allowed us to do is to be able to identify promising drugs for an emerging pathogen, in this case COVID-19, even though at the time of when this work was done, no successful and approved treatments for COVID-19 was available. Remember from the previous slide, the work that I'm describing was really taking part in the very early days of the pandemic between March 2020 and June 2020. And during that time, very little information was available. So how can one then even train on a machine learning model to identify candidate drugs for these novel emerging pathogens if there are no previous examples of successful treatments available? With that, all prior standard supervised learning techniques simply fail. And so the solution to that are few-shot learning or self-supervised strategies. And so with graph-based few-shot learning, what we were able to do is the following. We trained our model in 2 stages, where the first is called to meet the training stage. And during that time, we designed a large number of challenges to the model. And we asked the model to go through a large number of known human diseases that have some treatments and train the model such that it would be able to distinguish between drugs that are approved for those diseases and drugs that are not approved for those diseases. And we did that for many diseases. Here's shown on the slide, example for 2 diseases, but we did that for thousands of them. And so that was that stage finished, we then transferred the model to the second stage, which is meta-testing phase. And during that time, the model was simply presented with the description of COVID-19 and asked to produce a ranked list of drugs based such that at the top of that ranked list of drugs would be drugs that the model considered most promising to be repurposed for COVID-19. And so that was possible to do with the few-shot learning algorithm and our deep graph AI approach. Taking that predicted list of drugs, we very quickly shared it with our collaborators from National Emerging Infectious Disease Laboratories in the United States, and they have experimentally screened drugs that were predicted by the model. So the results of that work showed that 918 compounds that were screened for efficacy against SARS-CoV-2, first in African monkey Vira E6 cells and then in human cells, showed that those drugs that were top ranked by the AI model were highly enriched for drugs that show efficacy in human cells. In particularly, we obtained a near 62% success rate from testing top-ranked drugs. In contrast, only 0.8% hit rate would be obtained if that experimental screening would not be guided by AI predictions. And this is exciting because it's an order of magnitude improvement, an order of magnitude better hit rate among top 100 drugs than what alternative approaches would allow us to do. Beyond that, we were interested in knowing how did those molecules that the model successfully predicted and that were experimentally confirmed look like? Is there anything common to them? How do they actually target now these disease perturbed, like COVID-19 perturbed networks? How do they actually work? So what we found is that 76 out of 77 drugs that the model predicted correctly and that were experimentally verified were such that they do not directly bind proteins that are targeted by the virus. Instead, those drugs rely on network-based actions where they target certain drugs in human cells, certain proteins in human cells, and those proteins interact with other proteins in a cascading manner that eventually reach the proteins that are directly targeted by the virus. So this indicates that these drugs rely on network-based action, and they could not be identified by alternative strategies for drug repurposing, such as molecular-based docking. We refer to this class of drugs as network drugs because networks are crucial for them to exert their therapeutic effect. How are we able to find out what is common to those drugs? So the way we did that is by relying on explainable models that can take an AI prediction and an AI model that makes that prediction, and then produce an explanation, a short, human-readable, condensed summary of how the model generated that explanation. So with some of the algorithms that we've been developing over the years, now we have the ability that our users can ask questions such as, will a form of statin treat hyperlipidemia? What is the disease treatment mechanism of treating hyperlipidemia? And then the model would return a small molecular network, a small subgraph of the entire proteome, including pathways that link a particular drug and disease that the human expert was interested in. And that small subgraph, it represents an explanation and the core piece of the data that the model used in order to make a particular prediction. And so that part of that condensed summary can be used then by the human experts to decide when to trust an AI prediction and decide what are those predictions that are most actionable, most trustworthy, that can be used for downstream experimentation. So we actually wanted to take one step further and test that hypothesis, to what extent now AI-driven explanations are really useful to domain experts in identifying most promising hypotheses that AI models make. And we did that with collaboration with our partners from Mount Sinai in New York, where we compared our AI algorithm called Drug Explorer in terms of how AI-based explanations compare to settings where no explanations are provided, and what kind of effect those explanations have on user confidence, exploration time, and their ability to decide what are the predictions that they trust and that they feel confident about. And what we found is that through this user-centric study, providing explanations in addition to predictions really makes the model much more actionable, and experts have easier time deciding when to trust a particular prediction. In addition to identifying what drugs are most useful for what kind of diseases, we are also interested in modeling higher order or combinatorial effects that drugs might have in human body. So that refers to the problem of polytherapy, which is defined as a situation where patients take multiple drugs at the same time to treat complex or coexisting diseases. This is a really important problem. The statistics for US says that 46% of people over 65 years take more than 5 drugs at the same time. Many take more than 20 drugs to treat heart diseases, depression, or cancer. And that would be great. However, a patient that takes multiple drugs at the same time is at much higher risk for developing unwanted side effects. And 50% of population in US alone is affected by those unwanted side effects that are due to interactions between drugs. And that's a problem both for individual as well as the entire healthcare system, because it now means that those side effects need to be treated and further complicate the health status of an individual. So to model this problem, we teamed up with FDA, which is the regulatory agency for drugs in the US. And we did that to build a very large polytherapy knowledge graph, which is a graph of drugs and how those drugs work by targeting specific proteins in human body. So that graph had as nodes drugs, represented here by green triangles. Green triangles are connected by edges and indicating that a pair of drugs, when taken together, can lead to a particular kind of side effect, for example, RSAP1, and that side effect cannot be attributed to either drug alone. So it's really some interactive synergistic effects happening between those two drugs. In addition to information about drugs, we consider therapeutic actions of drugs in the sense of what are their nominal targets and how those targets interact with each other in the human body. And so that gives rise to a polytherapy knowledge graph, which when built, we can start using our graph neural network algorithm, which is called Decagon AI, that can answer queries about possible synergistic effects of drugs. So users can ask, for example, how likely it is that a pair of statin and ciprofloxacin, when taken together, would lead to breakdown of muscle tissue. And when that is being asked, what happens in the back end, the Decagon AI model would predict probability that an edge of type breakdown of muscle tissue would exist between those two triangle nodes that represent simvastatin and ciprofloxacin. And this is done in the context of the entire polytherapy knowledge graph. So the model can transfer information from other drugs and other proteins when answering this particular query. We validated predicted polypharmacy side effects by Decagon AI model against drug interaction markers, lab values, and several other surrogates, including standard drug interaction checkers for rheumatoid arthritis, IBD, Crohn's disease, at several hospitals in the US, and the results were very promising. They were so promising that we also did a follow-up study where we were interested in modeling adverse events for patient groups. So rather than looking at the entire patient population in the US as a homogeneous group, which is a very limiting assumption, we wanted to move closer towards precision medicine, where we can disentangle adverse events of drugs from the specific demographic properties and characteristic of a patient that takes that drugs. So we again teamed up with the US FDA and examined over 10 million reports of adverse events for the entire US population, covered over 19,000 distinct adverse events. And in doing so, we were able to identify then age and gender specific adverse events to identify what are groups of patients that are at lower or higher risk of developing an adverse event for a certain drug. So finally, let me conclude with a question and also offer one solution to answer that question, which is that really, how can we advance more broadly machine learning and AI for applications in science and medicine? And so I feel that that's really important question, because very often there are problems in biology and medicine, and our efforts in understanding disease biology comes with its own set of challenges that are different from other uses of AI. And so that means that it's important to be able to identify what those challenges are and then develop algorithms and methods that can address those challenges effectively. To this end, we have recently founded Therapeutics Data Commons. Therapeutics Data Commons provides a machine learning foundations for therapeutic sciences. It serves as a meeting point between domain scientists, such as biologists, chemists, medicinal chemists, who are identifying and formulating meaningful problems in biology and drug discovery as solvable AI tasks. And on the other end, machine learning scientists who are then taking those tasks and designing powerful AI ML methods. And so Therapeutics Data Commons support the development of novel AI ML theory and methods with a strong bent on understanding and developing the foundations of what algorithms are most suitable for drug discovery applications and why. And it is an open science initiative available at tdcommons.ai. And we're very excited that with TDC, we're able to cover an entire life cycle of therapeutics machine learning that starts from formulating meaningful machine learning tasks, then identifying, curating, and processing data set, developing powerful machine learning models in a variety of machine learning frameworks such as PyTorch, TensorFlow, JAX, Hugging Face, and so on, evaluating realistic model performance for those models, and then deciding which out of many models that might exist for a given problem is most trustworthy and ready to transition to the next stage of biomedical and clinical implementation. And TD Commons can do that by providing a very rich ecosystem of AI ML ready data sets, machine learning tasks, data functions, evaluator functions, and public leaderboards where members of community can submit their own models and predictions and allow us to identify best-in-class methods for a particular problem. Datasets in TDC cover a range of therapeutic modalities, including small molecules, biologics, antibodies, peptides, minerals, and genome editing therapeutics. And it's in the number of therapeutic modalities covered is increasing rapidly as we are continuing to grow Therapeutics Data Commons, both in the context of therapeutic modalities as well as stages of drug discovery and the kinds of biological questions that arise along the way. So finally, to conclude, Therapeutics Data Commons is an open science initiative, and it is really designed with our goal in mind, which is that of how can we advance machine learning and AI method development and really thoughtfully identify problems at the cusp of benefiting from machine learning, and then solving those problems and providing machine learning solutions in order to impact the future of biomedical research at the very fundamental level.
Great, thank you very much for the introduction. It's really excited to join this exciting event. So I'm going to share my screen. Okay, can you see my slides?
Yes.
Great. Yes, so I'm excited to share with you some of the recent research we've been doing using real-world data and AI to improve cancer trials and also to improve cancer precision medicine. So clinical trials is really the gatekeeper and also the bottleneck of all of medicine, right? It could be hugely expensive, right? So in the US, a clinical trial could actually cost hundreds of millions of dollars. And you can start with multiple, hundreds of drug candidates, and only one of them would actually make it through the clinical trial process, right? And because of this, right, the cost of actually bringing a new drug to patients on average could be several billions of dollars. Right. So this really motivated us to think about how can we use AI to make clinical trials more efficient and also make it more inclusive. Right. So for the first part, I will tell you a bit about this Trial Pathfinder, which is an AI-driven computational platform that we developed. It's described in this recent paper, and Trial Pathfinder is currently being used by Roche Genentech for designing to guide the design some of their new cancer trials. Right. And the idea of Trial Pathfinder is to really help to design the eligibility criteria of these trials. So the design of clinical trials is actually really complicated, right? So one reason why it's actually very challenging to design a clinical trial is that these typical trials have to decide which patients, right, are even eligible to participate in a given trial, right? So if you look at a typical cancer trial, they'll have pages and pages of PDFs, right, setting out rules to exclude patients from participating, right? If you have some lab values outside of some range, or if you have some previous conditions, then you're not eligible to participate. So these quite restrictive eligibility for clinical trials is really a major barrier for patients, especially for cancer patients. The majority of cancer patients are actually be happy to participate in clinical trials, because that's how often they can get access to some of the cutting edge treatment. But many of them are excluded, they're not eligible. On the other hand, right, so these trials are also, this also makes it quite difficult and challenging for the drug developers. A large number of cancer trials fail to reach the minimum enrollment, right, of patients because it's actually very hard to find eligible patients and recruit them. Also importantly, right, the fact that these clinical trials are very restrictive means that they're only testing the treatment, right? The clinical trial is only testing the treatment on a very narrow slice of the patient population, which is often the so-called Olympic athletes among the patients, right? And how well the treatment works on these Olympic athletes of the patients may not reflect how well it works actually on real world patients like you and me. So how does this happen? And then what's the current practice of designing clinical trials eligibility? So the ideal setting is that you want to design the eligibility of clinical trials, right? So that it protects the safety of the patients and also highlights the efficacy of the treatment. And unfortunately, right now we really lack data-driven approaches to help us to guide the designs of the trial eligibility. So here's actually some screenshots I took of an actual clinical trial, a cancer trial that's being designed, being developed. Right, so usually the first thing that people do is simply copy and paste over the protocols, right, from a previous trial that they did before. That seems pretty similar, right? And then they would go through line by line. Each line here corresponds to one of the rules that's used to exclude patients. All right, so for example, the first one looks at neutrophil count, right? And it says that if your neutrophil count is outside of some range, then you are not eligible. You're excluded from participating in this clinical trial. So the previous threshold is 1,500, right? So you can see actually seeing the comments in the Google Doc, right? So someone suggests, okay, can we actually relax this 1,500 threshold to 1,000 so we can enroll more patients, right? Somebody else would say that, oh, well, we don't really have evidence to change the threshold, so let's keep it to be 1,500 to be the same as before, just to be safe, right? And then there would be a similar discussion about hemoglobin count, right? So someone would suggest, okay, why don't we change it from 9 to 8 to enroll more patients? And there would also be a pushback that says, well, we don't really have evidence to change these thresholds. So then it ends up keeping it to be 9, which is what was used before. So the upshot of this is that you can see that the design of these trials is often based on heuristics, right? Not often based on data-driven approaches. And as a consequence, right? Even very similar drugs and similar clinical trials ends up using very different rules to exclude patients. As the illustration here, I'm showing you 10 different trials that are all looking at advanced non-small cell lung cancer. So each row here corresponds to one of these trials that was conducted to look at non-small cell lung cancer. Each column corresponds to one of the commonly used rules to exclude patients. Right? So some of these columns corresponds to different biomarkers, others corresponds to previous conditions and lab values. So I color-coded this to be a score to be yellow, right? If that particular trial ends up using that rule to exclude patients, otherwise it's colored gray. So you can already see quite visually, right, that there's even among these trials that are all looking at very similar drugs, some of them from the same company, right, they actually end up using very different rules to select patients. Right? So there's a lot of heterogeneity. And a lot of that is because of the fact that there's really lacks a data-driven and systematic approach for designing the eligibility of clinical trials. So this is really a huge problem. Right? So for example, in the US, the National Cancer Institute and other organizations have concluded and put out statements, right, that the eligibility of all the cancer trials needs to be greatly simplified and standardized in order to make it easier for diverse patients to enroll. But how to really design the eligibility and to select the eligible patients is really challenging, right? And this is where we want to help by using real-world data and using AI algorithms. So that's our motivation. So how do we do this, right? So I'm a computer scientist, right? So the key question here is really seems like a question about computations using counterfactuals, right? So effectively, we would like to answer the following counterfactual question, which is, I'll look at all these patients who were excluded from participating in the clinical trial, right? So which of these excluded patients would have actually benefited safely from the treatment? And our idea, which is quite intuitive, is that actually there's a lot of patients that end up using this treatment after it comes on the market, even if those patients were not eligible for the original clinical trial, right? Because the trial might be testing a very narrow slice of the population, Olympic athletes, but after the drug's approved by the FDA and after it comes on the market, right, then many people can actually end up taking the drugs. Sometimes them can even take it off label. So then the idea is that if we can actually identify these patients and follow them in their medical records, then we can see, okay, so what is the outcome of these patients from taking these treatments? Right? And to do this, we actually leverage data from the Flatiron Network, which is one of the largest cohorts of cancer patients, right? So it's based in the US. It has over 2 million cancer patients from all of the different states in the US, from over 280 community-based cancer clinics. And importantly, right, Flatiron actually has spent several hundreds of millions of dollars to curate and to clean this data set to make it very nice and well suitable for machine learning and data science applications. Right? So basically for each patient, we have their longitudinal outcomes for many years, as well as all of the medical records, their treatment, their outcomes, their lab values, etc. Okay, so then with this data, right, so the approach that we use with Trial Pathfinder is to basically use this very rich EHR data to computationally emulate clinical trials in silico. So here's basically how it works. I want to give you the high-level intuition. I suppose there's a particular trial that I'm interested in, right? So the trial would have like some treatment arm or some treatment drug, some control drug, which could be chemotherapy, and we'll have a set of rules for excluding patients. Right? In this example, you want to exclude patients based on bilirubin levels. Bilirubin is a common test for liver function. So let's say for this particular trial, then what we do is that we go into the medical records from Flatiron, right, from these millions of patients. Then we can computationally identify patients in the medical records who took the treatment, right? And then patients who took the control, right? And the patients who took the treatment, they also have to satisfy this threshold, right? The inclusion threshold of bilirubin less than 1, similarly with patients who took the control treatment. Right, so now this gives us 2 digital cohorts, right? The digital treatment cohort and digital control cohort. These 2 cohorts of patients might look very different, right? Maybe they have different demographics, etc. So then we can computationally match these cohorts, right, using machine learning and statistical approaches so that these cohorts are then comparable. Once they're comparable, then we can then sort of computationally emulate a clinical trial by computing things like hazard ratios for the outcomes from taking this treatment. So the power of this approach is that because it's done in silico, right, we can very easily actually just change the eligibility rules computationally. Right? So for example, I can say, okay, what if I relax the bilirubin threshold from 1 to 2, right, to enroll more patients? By doing so, right, then there are more patients in the medical records, then that satisfies this condition. Right? So my digital cohorts now becomes larger. But I can do the same idea of computationally match the treatment and control cohort, right? And then still look at the treatment outcomes. And the idea here is that by comparing this new in silico trial with the previous one, we can get a sense of like what is the actual impact of changing this bilirubin threshold, right? For example, if I change the bilirubin threshold and I get similar hazards, right? So then there's no more adverse events, then that gives me some data-driven confidence that perhaps I could relax the threshold to enroll more patients. So that's really the high-level idea. So here you might have a few questions, right? So one question you might ask is like, okay, so what if the drug is out on the market, right? Because for this approach to work, there has to be patients in the medical records who are already taking these drugs. But if there are drugs not on the market, then they will not be in the EHR. So to solve this, we actually use the fact that many of the drugs actually have very similar drugs, right, that have been on the market. Right? So for example, maybe a drug is previously approved for some other indications, and now it's being tested in clinical trial for a different indication, right? Or for a new drug, right, there might have been previous drugs that have similar mechanisms of action that were previously approved. A second question you might have, right, is that, oh, here we're working with observational data, right, which could have unobserved confounders. So this is the limitation to the study, to the approach. And we actually benefit quite a bit because we have very highly detailed medical records from many years and also have all of these lab values, right? And genetics data, which I'll mention in a bit. So this actually allows us to control for many of the confounders. And in addition, we validate our results actually using data from actual randomized clinical trials. So then to summarize, right, so the trial pattern approach then would take a given clinical trial, right? So it would do some sort of natural language processing to extract the relevant information, and then goes into medical records to identify these digital cohorts, and then estimate and sort of emulate the clinical trial in silico. And then it will do this millions of times in parallel, right? Each time it does this, it actually looks for different combinations of eligibility rules when it does like a new trial emulation. And in the end, this summarizes all of this information from these millions of in silico clinical trials, from millions of computational clinical trials, into one set of data-driven recommendations for what eligibility to set. So as a concrete example of this, right, let's look at this bilirubin threshold again, which again is a test for liver function. So here we're looking at 10 different clinical trials. So looking at immunotherapy for advanced non-small cell lung cancer. Each curve here corresponds to one of these clinical trials. And the x-axis corresponds to the bilirubin threshold. So the trial has some original threshold based on bilirubin for including or excluding patients. And we can computationally in silico, right, vary the bilirubin threshold while holding all of the other factors constant. And the y-axis here basically corresponds to the hazard ratio, right? So what's interesting here is that basically the hazard does not really change, at least when we vary the bilirubin from the more restrictive to the less restrictive, at least within this range. And that's a good sign because this suggests that we could actually make the bilirubin threshold less restrictive, right? And then still that enables us to enroll more patients without actually incurring more hazard or more adverse events. And we're able to validate these results using analysis from actual clinical trial, randomized clinical trial data. So at the end, right, so the trial pathfinder basically provides a set of data-driven recommendations for each trial, right, of what are the reasonable eligibility criteria to use. And of course, this becomes like a useful guideline and resource for the clinical teams when they design the clinical trials. We apply this approach to a variety of cancer types, both lung cancer, also to melanoma, colorectal cancer, and others. So here I'm showing you application of this to 10 clinical trials looking at advanced non-small cell lung cancer. Right, maybe the most important row is the last one at the bottom. Right, so the first 3 columns corresponds to, if we use the original clinical trial design, right? Here's how many patients would be eligible in each of these trials on average, right? So that's just about 1,500 here, and then this is the hazard ratio. The last three columns corresponds to if we use this data-driven criteria, right, which came from Trial Pathfinder, we can, on average, more than double the number of eligible patients by using our data-driven approach. And very interestingly, at the same time, we can actually even further reduce the hazard ratio. Whereas hazard ratio, lower the better, because hazard is bad, right? So this actually means that a lot of the patients who were systematically excluded from the original clinical trials, they would have actually benefited from the treatment to a similar or even greater extent compared to the very narrow slice of the population that were eligible. And critically, by taking our data-driven approach, right, that enlarges the patient pool, we actually enable a lot more women, more minorities, and more older patients to enroll and to access clinical trials. Right? Many of these patients were previously excluded systematically from being able to participate in cancer trials. So just to summarize this part, right? So we show Trial Pathfinder as sort of a first step, really in taking a data-driven approach using AI and real-world EHR data to help to make clinical trials more inclusive. And this is really a win-win, right? So it's a win for the patients because now they can enroll in the trials, and especially for the more diverse patients who were previously excluded, now they're able to enroll in these trials. It's also a win for the drug developers because this makes it it's easier for them to recruit patients and faster and cheaper to run these trials. We validated our computational approach using data from actual clinical trial data, right, from randomized trials, as well as data from other cohorts outside of Flatiron. Here we focus on cancer, but our methodology can be extended to other diseases. And we're very happy to see that this Trial Pathfinder algorithm is now being adopted by the pharma industry, by Roche, Genentech, and was also recently recognized as one of the top data science innovations of 2021 by the entire pharma industry. So in the last part, I want to talk to you about some quite recent extensions of this work in improving precision medicine for cancer. Right? Because one of the big challenges of cancer is it's very heterogeneous. Right? So two different lung cancer patients might have very different treatment disease trajectories. So to address this, right, we linked the data from Flatiron, which has these 2 million cancer patients, with information from Foundation Medicine, which provides sequencing-based mutation profiling of cancer patients. Right? By linking them together, we actually have over 40,000 cancer patients where we have their mutation profiles from NGS-based sequencing before the start of treatment. And then we also have their treatment outcomes, which treatment they're given, and the outcomes and lab values from each of those treatments from their medical records. And this enables us to really identify really interesting gene, mutation, and drug interactions. So here's an example of the kind of interactions. So for example, APC is like a tumor suppressor gene, right? And we're looking at bladder cancer patients who are taking immunotherapy. And we can identify in our data, right, that among those bladder cancer patients on immunotherapy, the patients that have mutations in this APC gene, right, they actually have much better survival outcomes. That's the orange curve. They have much better survival outcome compared to patients without the APC mutation who are on immunotherapy. Right, so this suggests that there's actually a positive interaction between mutations in APC and treatment outcome, patient outcome from immunotherapy in these bladder cancer patients. We can also answer the following complementary question, which is that among these patients who have all the patients that have APC mutations and with bladder cancer, right, is immunotherapy actually the best treatment for them? And in this case, the data suggests that it is the optimal treatment, right? Because among all these patients with APC, right, their outcome with immunotherapy is better, right, than their outcome than when taking other treatments. So mathematically, this corresponds to computation identifying this interaction effect, right, between the genotype, which in this case the mutations in APC gene, and the treatment immunotherapy. So, and in this particular case, we're able to validate our results, right? So we discovered this mutation treatment interaction effect using data from the observational data, but we're able to validate this using data from actually randomized clinical trials, right? So our collaborators at Roche actually run a clinical trial on bladder cancer patients on atezo, which is an immunotherapy. And we have also have mutation profiling data from each of those patients in the clinical trial, right? And in this clinical trial, randomized trial, we were able to validate our results, right? That APC, there's a positive APC immunotherapy interaction, right? So patients that have APC mutations who are given immunotherapy actually have much lower hazard ratios compared to the other patients. And this provides like a powerful predictive biomarker now, because now we can actually use APC as a biomarker to identify which patients like to benefit from immunotherapy, which is really one of the big open questions for precision cancer. So the example of APC is just one example, right? With our data-driven approach, we're actually able to identify over 450 such strong mutation treatment interactions, right, across many different cancer types. Right? So this is very powerful because previously in all of the literature, there were only a handful, maybe less than 60 such mutation treatment interactions that were previously known and from human clinical data. Right? And our analysis is able to basically increase this by an order of magnitude. So here's a visualization, a summary of some of these results that we have, right? So here I'm breaking it down by different cancer types. You have lung cancer, colorectal, breast cancer. Within each cancer type, we have different treatment classes, or immunotherapy, targeted therapy, right? So the color of each gene indicates whether, if it's red, that means that there's a negative interaction between that mutations in that gene and the treatment, right? So for example, if you have EGFR mutations, that means that you're less likely to respond to immunotherapy in lung cancer patients. And blue here means there's a positive interaction, right? So that means that, if, for example, if you have mutations in KRAS, then you are more likely to respond. You have better survival if you're given immunotherapy than if you have lung cancer. Right? And all of these, each of these genes then provides like a useful predictive biomarker to guide personalized treatment recommendations. In addition, we're able to also identify a large number of interesting mutation-mutation interactions. Right? So this means that if you have a patient that have multiple mutations simultaneously, we can identify, like, now, do these mutations have synergistic or destructive interactions or interferences? Right? And this is quite useful because there are several targeted therapies that are targeting specific mutations. For example, there are targeted therapies for ALK translocation. And we find that if you have ALK translocation and in addition, the patient has TP53 mutations, then these ALK targeted therapies actually will do much worse, right? Because of these mutation-mutation interactions. So there's a lot of information that we discovered here, right? And we actually provide a general new resource. It's called precisioncancer.org, where we have released all of this information. So you can find all of these mutation interactions, also mutation treatment interactions. To release all of this as a general resource for the cancer community. So just to summarize, right? So here, throughout both the projects, right? The main idea is that we can use AI and high quality real world data basically to computationally emulate expensive clinical trials in silico, right? And we have validated our results using data from actual randomized clinical trials, right? And the power of our approach is now is that we can really leverage the large scale data and the algorithms to do many of these in silico trials in parallel. I think there are many exciting applications of this. I just mentioned two of them, right? One application of this approach, as we saw, is to use this to take a data-driven designs of clinical trials to make clinical trials more diverse and more inclusive. And this enables more diverse patients to participate in the trials. It also enables us to discover new predictive biomarkers or mutations that can actually predict treatment outcomes for specific immunotherapies and targeted therapies. And there are many other applications that we're exploring based on this approach. So I just want to, the references are listed here, and I want to acknowledge my great collaborators, especially Rishan Liu, who is a fantastic PhD student in my group, who led both of these projects, as well as our collaborators at Genentech and Stanford. So I'll stop here. I'm happy to take any questions.
Good morning, everyone. Welcome to my talk. Thank you so much for Cell Press to get opportunity for me to be here to record my presentation, and obviously apologies for not being able to present online in person because of the time differences. So my name is Tingting Zhu. I am a Royal Academy of Engineering Research Fellow. I am part of the Institute of Biomedical Engineering that belongs to the Department of Engineering Science. So I am the IBME, Institute of Biomedical Engineering, as you can see on the pictures on the right, where here, if you can see my mouse cursor, is what we call the green building. I am part of the computational health informatics labs, which interestingly, the Tsai lab, which had its second site at Oxford Center for Advanced Research based in Suzhou, China. So our collaborators is quite broad. So we include worldwide leading clinicians from the local Oxford University Hospital NHS Foundation Trust, and also the George Institute for Global Health, also other developing regions such like Vietnam, Philippines, places in Beijing, Shanghai, Guangzhou, Hong Kong, and other locations across China. So today's my talk is about towards digital twin and healthcare. And most importantly, I would like to introduce the way how can we do personalized modeling, how to perform phenotyping, as well as risk prediction, and those things will then allow us to move step forward to go to the era of digital twin in healthcare. Right, so what do we do as a lab? I think this is always a question about. So the Tsai lab, in the logo is on the bottom left. We basically do not do imaging. That's pretty much it. We do basic non-parametric methods, deep learning. So we're in a lab that tend to apply state-of-the-art machine learning techniques and translate that to clinical practice. So we work with a lot of vital science labs, genomics, diagnosis medications, and also using wearable devices. So, internal personalization, the first thing I'd like to introduce is about how do we able to provide dynamic forecasting of patient health in those for critical care. The current challenge, imagine that we now have Fitbit and wearable devices that we can allow us to collect data on a daily basis. So the question is not about how much data can we collect, and it's actually the challenge is the quality of data. What am I showing you here is just blood pressure data, systolic, diastolic, and mean arterial blood pressure. What you're seeing here, if you can see my mouse cursor, is that you got a lot of artifacts, and then you got disconnections of the sensor, as you can see it on here. And then, so it's very noisy, right? So it's actually quite challenging is to overcome that problem when you start having large amount of data and they're very noisy and you don't know which one is of good signal quality. So wouldn't it be great if we have sort of traditional way to model the data and give us some confidence about how noisy the data is, and also to give us imputation when there's missing values, right? So here's showing you the x-axis is hours during the patient stay. So SpO2 is just a measure of the oxygen in your blood. And what we have is the blue line is the sensor data, and the nurse observation will be every 4 to 6 hours and in circle. Idea is that we can introduce something called Gaussian process as a way to impute the missing value, as well as giving us a strong confidence bounds about, when it's missing, you can see that the confidence bound is much bigger, so it's less uncertain, where then it's not missing is very narrow. So what can we do with that? Isn't that would be great, right? So the problem in real life is that then that add on to another level about fitting the model. So here I'm showing you the concept of just modeling heart rate that change over time. So the top one is about what we want. So ideally that we want to fit a model such as Gaussian process in this case among the dotted data point, and then we want to forecast, want to predict in the future about what happened to that individual. But what you end up having most likely is something what we call underfits, meaning that your model is not fitting your data perfectly, or overfitting, in this case that you are literally fitting every single data point of the data, and then you will provide very low predictive power. So what in our lab have come up with is something called the Bayesian dynamic forecasting of vital signs. Here I'm about to show you an animation how it's done. So in the x-axis, the time of warding patient state, and then y-axis as an exemplar, very simple for visualization. I'm showing you the heart rate beat per minute. So what you have is a current window, what we basically fit a Bayesian Gaussian process in, and then the Gaussian process is going to sort of learn what are the data are useful. As you can see that it would actually ignore some of the artifacts. And then it's trying to forecast, which is in yellow, of what it believes the mean value as the bolded in black line, and what would be the confidence around what those values should lie as the future heart rate. So why does it matter? So if we play the video, as you can see, the Gaussian process is actually adapting in real time and trying to forecast ahead. As you can see that the data roughly fall within what the Gaussian process believes in. Up until a point, which is about to happen, is that you can start seeing that, well, so all of a sudden, so what you are seeing here is actually the fact that the Gaussian process is actually expecting the heart rate, essentially, if you looked at it, is the mean value around 130 of that individual. And so it forecasts around 130. But in real life, what you observe is actually the patient heart rate is going down by 10, 20 beats per minute. And what essentially we could use that is that what we've learned is about more personalized. Ideally, is that if we model the patient's vital sign, in this case, heart rate, that change over time, and then we can forecast what will be the norm and expected value of that individual. And then then we can compare what it actually observed differently. So from there, in this case, actually the patient was gone through a deterioration actually and dying because of it. So despite the fact that you think 120 is normal, when you reduce your heart rate, the lower the better, but actually for that individual, it actually has some sort of deterioration happening. So using a way of longitudinally Gaussian process modeling, then we could actually provide by means of forecasting something that was norm, and then we can identify outlier, abnormality of that individual. So, okay, what other thing can we do with Gaussian process? My student now, who is a doctor, assistant professor at NYU Abu Dhabi, Farah Shamu, she also used Gaussian process for imputation of missing value. As you know that in electronic health records, there's a lot of data are missing and data are collected unevenly and large amount of missing data. So she compares different way of imputing missing values, such as last very careful linear interpolation or using Bayesian Gaussian regression. So then once she imputed the value, she'll feed that into a neural network, in this case, recurrent neural network, and also she was one of the few that actually used attention to indicate for early warning scores, meaning that basically you have a trajectory and then you have some sort of outcome within the next 24 hours. You want to predict if that patient is going to experience some sort of event, and you want to alert clinician 24 hours in advance. So visually showing you a diagram here is that just showing you the general vital sign, what we normally observe in a hospital stay, and then you see that there's outcome, and then we're basically able to use attention map, in this case, to identify the importance of value that indicate to, that refers back to deteriorations. Great. Okay, so moving on from there, then how do we do more personalized health trajectory modeling then? Great, now let's move on to more personalized health trajectories. Imagine if we've got very long trajectory of individual that change over time, or maybe even repeated treatments of an individual. So what I mean by that, so imagine that you have systolic blood pressure, as I just mentioned, of multiple treatments, right? So we're modeling a blood pressure treatment that changes over time in multiple treatment sessions. And each time, if you looked at it, they're very different, right? So the dotted black lines patterns is very different in each treatment visit. And sometimes here you actually have missing values. So the concept is basically that we got some noisy or observational data of that individual for each treatment. And then we want to infer something latent, something that is unknown, that is underlying how latent trajectory of that individual. So in that case, you will have section 1, 2, and 3 as an example, which is a noisy version of the latent health trajectory, and we want to infer something in red that is unknown for that individual. Here, I'm going to show you a case study about a complex disease, which is chronic kidney disease. So CKD is actually affecting 5 to 10% of all populations. It's in the UK, the prevalence of end-stage renal failure is 861 per million, and 50% of those actually undergoing hemodialysis. Hemodialysis is basically the concept of cleaning the waste in your blood, essentially. So the problem about HD is actually it's quite general symptoms. The patient generally feel unwell, it's got nausea, vomiting, and most important, they have something called an interdialectic hypotension. That is a sudden drop of blood pressure. So the challenge here is that how do we understand, how can we predict the sudden drop of blood pressure and provide early warning of that? Once your blood pressures drop, it's quite straightforward. If you put your legs up, you lie down, then you balance the blood pressure, then you can avoid all these nasty outcomes. Because of IDH, that actually patient survival rate is quite low, right? Going through hemodialysis, the first is 80%, in year 268, and then by 10 years, you have 1 in 5 survival. So what you normally see as a nurse dialysis monitoring is basically you're going through some blood pressures and vitals and stuff, and you see in real time that. And what you want to do is model those trajectory that is changing over time, that then allow you to infer the latent health trajectory of that individual. So a bit more what I just talked about. So you got repeat treatments and multiple treatments of the same individual. In this case, just simplify showing you blood pressure. And then what you want is learn the latent trajectory of that individual. So it turned out that you can do that through something called the hierarchical Gaussian processes. So ideally, you can, top right here, I'm just showing you in a graphical model, what you can visualize is as, the latent trajectory is actually the parent trajectory, the parent structure that you want to learn. But what you observe is the children structure. So the child structures are the repeated measurement from clinical visit or treatment. And then so the concept is like we are mutated from our parent, right? So we are sort of a variance of the parent. So we all derive from the same parent in that case. Yeah, so what we're seeing here is essentially that you got children, which is a variant of the parents. So sort of mutated from our original parent. Parent is something what we learn is the latent health trajectory. So from there we can learn something what is normal. We also can learn something that is abnormal. Okay, so what do we do with that? Once we learn the healthy latent trajectory, which what we call the latent Gaussian processes representing the normal behavior, so we can actually collide them into a dictionary. Once we have that dictionary of healthy trajectory for each individual, then we can do simple template matching, right? So when the new pattern comes in, we can fit it with a Gaussian process, and then we'll do KL divergence to compare the similarity between the two. So what I'm showing you here is an animation about what do I mean by that. So it's essentially you take a blood pressure pattern, in this case, that is a segment, and then you call that a new pattern. And then you have a dictionary of healthy patient trajectories that you can compare with. And then what we have is something called the novelty score. The novelty score means is how new, how unfamiliar you are compared to our dictionary. So this new blood pressure pattern then is compared to our novelty, our dictionary to compute the novelty score. As you can see, high, medium, high, and low. So by that, we can then identify outliers and something is very different from what we normally see. So then we can call that is a normal pattern and alert the clinicians. So essentially what we have is that we can do is stratify the reference segment by time within the sessions, or even a particular session within a week, or you can have a patient cohort or personalized to that individual from previous sections. So you got time series data comes in, and then you have this sort of template matching with the new patterns, with Bayesian Gaussian GP, Gaussian process, and then that compared to a dictionary, and then you have a detection abnormality. So can we go deeper? Because we're in the era of deep learning. The answer is yes. So imagine you've got multiple sections, say section 1 to K, of the individual subject 1, and you've got multiple subjects. You can actually add layer to it, and that form clusters of subjects. So you have normal versus abnormal class of subjects. You've got different class of abnormality, and then also different class of normal populations. So here I'm just showing you a diagram. What do I mean by that? So the top right is number of subjects, and you can see that some people exhibit some sort of trajectory of blood pressure, and some others, much lower, or so they have different profile and concept. So that's why it's deep time series clustering using trajectories. Okay, so now I demonstrate to you that with something simple like blood pressure that changes over time, we can start clustering the individual. But what if we want to identify phenotypes? What if we have heterogeneous data type? We've got different data, in electronic health record, it's not just continuous value, we have some information about individual. And we want to understand the outcome of those individuals. So what do I mean? So imagine you've got all different various data type coming into the diagram here, and what you want to measure, and what you see here is sort of the trajectory. Right here, I'm just showing you some systolic blood pressure or respiratory rate as continuous value, even see CRP as blood. As you can see, it's very discretized because blood does not collect every hour or so. It's quite on a daily basis, or even sometimes within the interval of a couple of days. And then you also have background information, static, like type of admissions and what kind of event it is. And then at the end of it, you want to phenotype that. You want to cluster to understand, there's different type of individual or subpopulation, and then you want to understand how can we use it to predict, say, mortality, disease progressions, how does it respond to treatment, or simple as what I just described, abnormality prediction. So my student, Henry K, is currently actually working on something pretty cool called the deep phenotypic time series clustering. It's recently been accepted to ICML 2022. So for those who are interested, please do contact me for the model. So ideally, this concept is that basically we have all of this high-dimensional complex heterogeneous informations, and we want to squash it in through a recurrent encoder to give us a latent representation Z. And then from there, we have an identifying network to identify the cluster assignment probability. And most importantly for this model, it's different from a traditional clustering algorithm, is that we all know clustering generally is unsupervised, meaning that you sort of difficult to validate the number of clusters. But in this case, what we propose is a supervised clustering methodology. So meaning that we are able to use the outcome that we're interested in. In this case, will be death, ICU, cardiac arrest, or discharge. That is in a hospitalized setting, 24 hours in advance. And we are able to use those labels to train and guide us through the clustering process. So what is the advantage of that? That means that we can then, in our validation, in our test set, then we can actually tell how correct is our cluster when we're assigning individual to a particular cluster group, and whether that actually gives us a correct prediction of the outcomes in the future. So what I'm showing you on top right here is that ideally you have, if I just look at different group of people, as it is, and look at different variables, as you can see, they're all tangled together. You can't really just say draw a line and say, well, heart rate value for people who have ICU admission is here, and then healthy individual is somewhere else. It's very difficult to untangle them. But with the clustering approach, we're able to actually provide a separate, as you can see at the bottom, a clear trajectory of different subgroups. So what we're showing here is 6 groups, and we call them unique phenotypes. So for what do I mean by phenotype in this case? So phenotype here is actually means is a probability distribution of the outcome of interest, right? So I got 6 clusters, each of them represent a unique representation of phenotype, meaning that in this case, I will have a probability, a phenotype of probably a high no event individual and 0 deaths, that 0 cardio and almost no death, and also ICU. So I'm start understanding certain classes are cardiac cluster, they have their unique phenotype, or I have ICU with healthy, or also have classes that have death or severe ICU patients. More importantly, for this deep phenotypic clustering method is that it allows us to understand how variable changes over time and then that leading to an outcome. So what do I mean by that? As you can see on the diagram here, these are cluster-based attentions, meaning that we have individual attention, so we know each individual patient, how they change over time in relation to variable. So here just a sample of sets of variable. Obviously we have longer and more, it's just harder to visualize. But what I want to draw attention is that you will also able to find a cluster-based attention. So in that subpopulation, that in that particular phenotype, what type of variable or which type of variable are most indicative and important. So the brighter color is more important, and whether they change over time. So in this case, FiO2 for this particular cluster, it's very important. Well, in a different cluster that actually gender play an important role, right? So that is really exciting. How do we use that in practice? So essentially, you have patient being observed by clinician or by nurse that passing through the deep phenotypic cluster, and then in that cluster we will generate cluster phenotypes. And from the phenotypes, we can actually understand what variables are important, so we can make recommendation for action. So, and it's explainable because we can tell which variable at what time it makes a contribution to the outcome of that happen in a future event in, say, 24 hours in advance or 6 months down the line. So we know that we can make a recommendation, more action, what would be the next relevant action to take. And from that, we can then feedback with additional symptom information back to the patients and going through the phenotype that then reclassify where that individual is. So that is a change over time in a dynamic sense. Okay, so far I have talked about personalized monitoring and template matching, and also learning personalized latent trajectory, and also phenotyping. The next thing is about how do we utilize all of these towards building a digital twin. So digital twin is a buzzword, I must admit, that people talked about a lot. And there isn't any particular example existed to say, well, can you create a twin version of an individual? Why does it matter? Because imagine in the world that if we able to create a virtual copy of us, then we don't actually need to take bloods, don't need to actually get data, but we can use variable sensors or any information, or we can even do something called generating electronic patient information, and then the clinician can make a diagnosis based on our digital twin, right? And then also that will help us inform effective treatment down the line. So here's essentially what we want is that we, first of all, the bottleneck is how can we generate real patient information. So ideally that we want to feed real patient information, as you can see here. We want to generate but fake but realistic information of the individual, that top right. So my student Jin, she had actually just created this very interesting model about generating outcome-specific medical information. The particular interest for this model in compared to other models in the literature is that a lot of people specialize in just generating continuous values, vital signs of data. Some people just generate very discretized intervention data. And there hasn't been any work done on generating both. And if you think about it, it's quite difficult in a sense that, right, because if we are male, female, and then, you know, can you learn your data for vital signs and then try to map with some sort of binary values? But they are actually not binary. So in machine learning, we tend to just like put them in one hot encoding, we just arbitrarily set them to 0 and 1s, but they are not 0 and 1, they actually mean something. So how do we actually learn this information? How do we learn the correlation among each other in a later representative? So what we've done here is that we model them separately from the raw space for our generator. So, and then we then say, well, they all come from the same individual. So in the later space representation, they should be very close to each other. That's the key novelty of this work. So for those who are not familiar with GAN, Generative Adversary Network, essentially you have a generator basically generating information that close enough to the real patient data, but not exactly the patient data. And then it passes through a discriminator. A discriminator then decides whether have you generate realistic patient information. If you do, then you pass the mark. If you not, then go back and learn some more. That's the general concept. So we've done some testing into results. For the interest of time, I'm going to rush through this. So we compare all the state-of-the-art models in the literature, and then we show that our results is much better, produce smaller mean absolute error. Also, we have a better accuracy in recall and positions. But if you know the outcome that you want to generate for specific patients, we can, in this case, that we showcase that we have four group of people, and we are able to separate them really well. Here is showing you the real correlation among different variables, and the right is what we simulated. And here's some example of the result in the raw time series, how they look like. So at last, I'd like to sort of conclude this. It's about, okay, we are in the path of going to the era of digital twin, but we're not there yet. But we have all essential tools to allow us to able to get closer and closer to create this digital version of us. And the last thing what I want to cover is something, the work that my student Dan and Danny for continued learning across disease and data. So a lot of machine learning algorithms, they have something what we call catastrophic failure, meaning that when you learn from one data set for one task, it's perfect. But then you're trying to use the same model to apply another data set, despite you do transfer learning or domain adaptation, it's going to degrade the performance. But unlike humans, for example, if I want to learn how to play basketball, and then I want to learn table tennis, it doesn't mean that I will forget how to play. I forgot how to play basketball, right? So that's what we're trying to mimic the learning of an algorithm here. So we want something called, the lifelong learning, the continual learning, meaning that we want to make sure the algorithm is learned continuously and mimicking how human behave and develop. So I'm showing you the top diagram here is that where you can see the catastrophic failure of the algorithm of AURC as it start learning a new task, right? And it's going down, up and down, depending on the task and then depending on the data set it has. But then using our proposed model, you can see that it actually quite maintaining the AUC of the previous task while learning new tasks as it go on that change over time. And this works between different diseases and also different data sets. So thank you very much for your time. Sorry again that I'm not online, but do please get in touch. Here's my email address if you're interested in any of the work that I'm doing. Thank you.